Hello, my name is Sashelle King, and today I will be presenting chromosome structure and variations. So, first, let's start off with a very basic question. So, what is a chromosome? Chromosomes are long, thread like structures found within the nucleus of eukaryotic cells. They play a very crucial role in organizing and carrying genetic information. So, here let's go over the basics of chromosomal structure. So, let's start with the DNA molecule. Chromosomes are primarily composed of DNA, which carries the genetic code for organisms. And we all know that DNA is a double-stranded molecule made up of nucleotide units. Now let's talk about chromatin. When not undergoing cell division, chromosomes are in a diffused and less condensed state that we know as chromatin. Chromatin consists of DNA wrapped around histone proteins. This arrangement allows for efficient packing and regulation of gene expression. We'll move on to chromatids. So, during cell division, specifically during S phase of the cell cycle, chromosomes replicate, resulting in two identical copies called sister chromatids. These sister chromatids are held together by a structure called the centromere. So, let's talk about the centromere. So the centromere is a specialized region of chromosomes that plays a very crucial role in chromosome segregation during cell division. It serves as an attachment point for spindle fibers, which move chromatids to opposite ends of the dividing cell. Now let's talk about the telomeres. So telomeres are protective structures found at both ends of a linear chromosome. They consist of repetitive DNA sequences and proteins. Telomeres help maintain the stability and integrity of chromosomes during cell division and protect the genetic information from degradation. Chromosomes are classified into various types based on their characteristics, size, and role in an organism's genetic makeup. We're going to go over some common types of chromosomes. So on this slide, we will talk about autosomes and sex chromosomes. Autosomes are non-sex chromosomes. So in humans, there are 22 pairs of autosomes. They carry genetic information related to general body traits and functions. Autosomal chromosomes are the same in males and females. Sex chromosomes, though, they determine an individual's biological sex. In humans, there are two types, X and Y. Males have one X and one Y, while females have two X chromosomes. On this slide, we will discuss heterosomes and homologous chromosomes. So heterosomes are another term for sex chromosomes, which include our X and Y chromosomes. Homologous chromosomes are pairs of chromosomes that have the same genes in the same order, although they may carry different alleles. One homologue is inherited from each parent. Homologous chromosomes are involved in genetic recombination during meiosis. Let's continue adding to our list of different chromosome types. This slide will cover non-homologous chromosomes and metacentric chromosomes. So, non-homologous chromosomes are chromosomes that do not belong to the same homologous pair. They carry genes for different traits and are not involved in genetic recombination with each other during meiosis. Our metacentric chromosomes have a centromere located near the center, dividing the chromosome into two roughly equal arms. Okay, so let's add two more to our growing list. We have our submetacentric chromosomes and our acrocentric chromosomes. So, submetacentric chromosomes have a centromere located slightly off center, resulting in one longer arm, the Q arm, and one shorter arm, the P arm. Acrocentric chromosomes have a centromere located close to one end, resulting in a longer Q arm and a very short P arm. In humans, the 5 pairs of acrocentric chromosomes include the ones carrying the genes for ribosomal RNA. Okay, so now we'll talk about our satellite chromosomes and our telocentric chromosomes. So, satellite chromosomes are a subtype of acrocentric chromosomes and are characterized by the presence of small bead-like structures, the satellite, attached to the Q arm. They are named for their appearance under a microscope. Telecentric chromosomes have their centromere at one end with one long Q arm and no P arm. These are relatively rare in humans. I've added a little picture so we can all see it because it's easier to see than to visualize with just words. Okay, so let's talk about our last two chromosomes on our list. We have our B chromosomes and our polyploid chromosomes. So B chromosomes, also known as supernumerary or accessory chromosomes, are additional chromosomes that may exist in some individuals of a species, but are not essential for the organism's viability or reproduction. They often carry non-essential genetic material. Polyploid organisms have multiple sets of chromosomes beyond the typical diploid number. For example, triploid organisms have 3 sets of chromosomes, tetraploid organisms have 4 sets, and so on. These types of chromosomes play various roles in organisms' genetic makeup and contribute to the diversity and complexity of genetics in different species. Now we'll dive into chromosome structure and variation. So here we'll talk about deletion, duplication, inversion, and translocation. These are 4 types of structural chromosome abnormalities that involve changes in the structure or arrangement of genetic material within chromosomes. These alterations can have significant consequences for an individual's health and may be associated with genetic disorders. Let's go over each of these and get an in-depth explanation. So, deletion. Deletion is a chromosomal abnormality in which a segment of a chromosome is missing or deleted. It results in the loss of genetic material. Deletions can occur due to errors during DNA replication, exposure to radiation or chemicals, or as a result of spontaneous genetic mutations. So, what are the consequences of these deletions? Deletions can lead to genetic disorders if they remove essential genes or regulatory elements. The severity of the effects depends on the size and location of the deleted segments. So now let's move on to duplication. So by definition, duplication is a chromosomal abnormality in which a segment of a chromosome is repeated, resulting in an extra copy of that genetic material. Duplication can occur as a result of unequal crossing over during meiosis, leading to an extra copy of a chromosome segment. Consequences of duplication can have a range of effects, from causing genetic disorders to increasing genetic diversity. If the duplicated segment contains genes, it can lead to a dosage-sensitive condition. For example, Charcot-Marie tooth disease is associated with gene duplications on specific chromosomes. Now let's talk about inversions. By definition, inversion is a chromosomal abnormality in which a segment of a chromosome is flipped or reversed in orientation within the same chromosome. Inversion can result from chromosomal breaks and rejoining in an inverted orientation during DNA repair process. Inversions may not always lead to genetic disorders, especially if they do not disrupt essential genes. However, they can affect gene expression and may have consequences during meiosis, potentially leading to infertility or an unbalanced number of gametes. Lastly, let's talk about translocation. So translocation is a chromosomal abnormality in which a segment of one chromosome breaks off and attaches to a non-homologous chromosome. There are 2 main types of translocation, reciprocal and Robertsonian. So reciprocal translocation is an exchange of segments between 2 different chromosomes. And the Robertsonian one is fusion of 2 acrocentric chromosomes that typically result in one large chromosome or one small fragment. Causes of translocation can occur spontaneously or be induced by radiation or chemicals. Translocations can result in balanced or unbalanced translocations. Balanced translocations involve an exchange of chromosome segments without loss of genetic material, often having no noticeable effects. Unbalanced translocations, on the other hand, can lead to genetic disorders if there is a gain or loss of genetic material. A well-known example is the Philadelphia chromosome associated with the chronic myeloid leukemia, CML. So our list of chromosome variations continue. On this slide, we'll go over ring chromosomes, isochromosomes, and aneuploidy. So these are important types of chromosomal abnormalities that involve changes in the number and structure of chromosomes. So let's talk about ring chromosomes. A ring chromosome is a structural chromosomal abnormality in which the ends of a linear chromosome fuse together to form a circular or ring-like structure. Ring chromosomes often result from chromosomal breaks and the fusion of those broken ends. The ring structure can be stable or unstable, with potential consequences for gene expression. Some consequences of ring chromosomes is that it can lead to genetic disorders, especially if they result in gene loss or disruption. The instability of ring structure can cause issues during cell division. Let's move on to isochromosomes. An isochromosome is a chromosomal abnormality in which one arm of the chromosome is duplicated and the other arm is lost. This results in a chromosome with two identical arms, either both long, QQ, or both short, PP. Isochromosomes can occur due to errors during cell division, particularly in meiosis. This can lead to genetic abnormalities if they disrupt gene dosage or affect essential genes. For example, an isochromosome of the long arm of the X chromosome can cause Turner syndrome. Last on this slide is aneuploidy. Aneuploidy is a chromosomal abnormality characterized by an abnormal number of chromosomes within a cell. It can involve the gain or loss of a whole chromosome or parts of the chromosome. This often results from errors during cell division, particularly during meiosis or mitosis. Trisomy, an extra chromosome, and monosomy, a missing chromosome, are common examples. Consequences of aneuploidy can lead to a range of genetic disorders, depending on which chromosomes, like Down syndrome, trisomy 21, which results in a copy of the chromosome 21, and Turner syndrome, which involves a single X chromosome in females. Okay, so this is our last slide on chromosome structure variation. We'll go over 4 more. We have polyploidy, mosaicism, ring chromatids, and fragmentation. So, polyploidy by definition is a chromosomal abnormality characterized by having more than 2 complete sets of chromosomes within a cell. So you can also have like triploidy or tetraploidy, and higher levels of ploidy can occur. Polyploidy can result from errors during cell division, typically in meiosis. It is relatively common in plants, but rare in animals. Polyploidy can have significant effects on an organism's phenotype, and it can lead to genetic diversity. It is typically lethal in humans, as extra sets of chromosomes disrupt normal development. So next, let's move on to mosaicism. Mosaicism is a genetic phenomenon in which an individual has 2 or more different cell populations with distinct chromosomes compositions within the same individual body. This typically arises from mutations or chromosomal abnormalities that occur during early embryonic development. These genetic changes may affect some cells, but not others, leading to presence of genetically distinct cell populations. The consequences can vary widely depending on which cells are affected and the nature of the genetic changes. Some cells may carry normal chromosomes, while others may have chromosomal abnormalities. Mosaicism can result in a range of clinical manifestations, from mild to severe, depending on the tissues and systems affected. It can complicate genetic diagnosis and counseling. Okay, let's move to our third one on our slide, the ring chromatid. So, by definition, a ring chromatid forms when a chromatid, one of the two identical copies of a chromosome during cell division, forms a ring structure. This can lead to genetic instability. Ring chromatids can arise due to chromosomal breaks. And these breaks and rearrangements can occur spontaneously or due to external factors. The presence of a ring chromatid can lead to genetic instability and may result in issues during cell division, including difficulties in chromosome segregation. The specific consequences can vary depending on which genes are affected and the stability of the ring structure. Lastly, let's talk about fragmentation. So, fragmentation refers to a chromosomal abnormality in which chromosomes break into two or more pieces, leading to loss of genetic material. This can result from various factors, including exposure to radiation, certain chemicals, or DNA replication errors. It can also be associated with underlying genetic instability. Some consequences of fragmentation can lead to the loss of genetic material. The effects on the individual depend on which genes or genetic regions are affected by breakage and loss. If essential genes are deleted, it can lead to genetic disorders or developmental delays. These chromosomal abnormalities highlight the genetic diversity and complexity observed in individuals and populations. Mosaicism, ring chromatids, fragmentation, polyploidy, and polyploidy can all have significant clinical implications, and they are studied in the field of cytogenetics to better understand their cause and effect on human health. Now let's go over some overall causes of chromosomal variations. So we have spontaneous mutations. So errors in DNA replication, repair, and recombination can lead to chromosomal variations. We can also see some environmental factors that come to play. So there's things like exposure to radiation, chemicals, or different toxins. This can also increase the risk of chromosomal abnormalities. Then, of course, there'll always be genetic factors. Some individuals may have genetic predispositions that make them more susceptible to chromosomal variations. And then, I didn't add this one on the list, but of course, advanced maternal age. So, the older a mother's age, specifically over 35 when having a baby, increases the risk of things like polyploidy and aneuploidy. These consequences of chromosomal variations can range from no noticeable effects to severe genetic disorders or developmental abnormalities. The impact depends on the specific chromosomes involved, the genes within those chromosomes, and the extent of the chromosomal alterations. So we've already touched on these, but these all have a variation of chromosomal numbers. So numerical chromosomal abnormalities can have significant consequences for an individual's health and development. The severity of these effects often depends on several factors, including which chromosomes are affected, the specific genes within those chromosomes, and the extent of the abnormality. These variations can lead to a wide range of genetic disorders, developmental abnormalities, and health challenges. Numerical chromosomal abnormalities are typically detected through genetic testing, such as karyotyping or chromosomal microarray analysis. Genetic counseling is often recommended for individuals and families affected by these abnormalities to provide information, support, and guidance regarding potential risk and management options.